सेशन में मैसेज कर दो।
तो देखिए बच्चों से वीडियो वाला क्या कितने लोग दिख रहे हैं
सर का इंट्रोडक्शन देता हूँ।
ठीक है
फिर आपको खड़े हो जाना है। फिर तो खड़े हो
मैं खड़ा हूँ उसको दिखता रहेगा ना जो बोलना है उनका इंट्रोडक्शन हाँ लिखिए
हम खड़े हो गए तो आप कर दो।
गुड मॉर्निंग सर देख पा रहे हैं
या वेरी गुड मॉर्निंग
हाँ
वेरी गुड मॉर्निंग
हाँ सर बस अभी हम लोग आपकी आवाज़ एकदम ऑडिबल है सर और क्लैरिटी है दिख भी आपको एकदम क्लियर दिख रहे हैं सर और होगा क्या आपको सिर्फ स्क्रीन दिख रही होगी लेकिन बच्चों को आपका पीपीटी की स्लाइड और उसके ऊपर आप भी दिख रहे होंगे सर एक छोटे से विंडो में आप दिखेंगे और पूरी विंडो में पीपीटी दिखेगा ठीक है ना सर तो थोड़ा सा और एक अच्छा वो होता है मतलब एग्जैक्टली सर 10:30 पे मतलब वी विल स्टार्ट विद योर इंट्रोडक्शन
ओके
हाँ मेरे इसमें है सर का व्हाट्सएप नंबर रक्तिम लिखिए RAKTIM डॉक्टर रक्तिम मुखर्जी ये इनका व्हाट्सएप नंबर है
सर लाइव चैट चालू कर दिया क्या बिकॉज
नहीं सर वो आप यूट्यूब का जो लिंक भेजा है ना
हाँ यूट्यूब में आ रहा है व्हाट एवर वी आर स्पीकिंग दैट इज आल्सो कमिंग राइट नाउ
यूट्यूब पे लाइव जा रहा है क्या ओके
हाँ लाइव जा रहा है दैट इज
ओके सर अच्छा सो आई विल स्विच ऑफ माय माइक यस सर
इंट्रोडक्शन आप ही दो जो सर वो
10:20 थे अच्छा 10:30 में छोकरा टाइम
आप ही को मांगो चीज
એવું ના ચાલે સેશન તો
लाइव में इनके डिले है सर
अच्छा હું બીજી વસ્તુ કરતો રહીશ
लाइव में डिले है सर
ઓકે So Raktim sir,
Yeah, tell.
should we start, sir?
Yes, you can.
So dear participants, good morning to one and all. We are about to start the fifth lecture of our e-crash workshop, June 2020, conducted by Network of Biotechnology Capacity Building Cells of Vallabh Vidyanagar Unit of Gujarat State Biotechnology Mission and N.V. Patel College of Pure and Applied Sciences. As you all well aware that today we are having the session by Dr. Raktim Mukherjee on Introduction to Animal Cell Culture. Before proceeding, let me give introduction of Dr. Raktim Mukherjee. Dr. Raktim Mukherjee is currently head at Sri P.M. Patel Institute of PG Studies and Research in Science, which is affiliated to Sardar Patel University. Dr. Mukherjee has completed his PhD or doctoral degree from Department of Zoology, MS University of Baroda, under the renowned Professor A.V. Ramachandran. He is having 20 publications to his credit in national and international journals. He did his graduations and masters both from MS University of Baroda, and he is involved in teaching subjects like immunology, human physiology, animal biotechnology, development biology and reproductive medicine. Sir has presented more than 40 papers in national and international conferences and also won several best paper awards. He is involved as a speaker in so many of the crash workshops conducted by GSBTM. He is also the member of Society for Reproductive Biology and Comparative Endocrinology, Chennai, India. He is a member of Indian Physiological Society. He is member of Indian Immunological Society. The most notable thing about Dr. Raktim Mukherjee is that he is a quiz master and taking quiz is his passion. So he is involved in professional quizzing since last 22 years, and there are so many. If you will search on YouTube and other channels, you will find so many quizzes which are being conducted by Dr. Mukherjee at various platforms, including GI Bion of the Gujarat State Biotechnology Mission. Besides that, Reliance Inter-School Quiz, Lions Club, Bank of Baroda Employees Quiz, All Baroda Inter-School Quiz, and many more. He has also, his quizzes are very popular among students, and he has also took these quizzes at Sardar Patel University Interdepartmental Quiz, Faculty of Science of MS University of Baroda. In a nutshell, we are very much fortunate and privileged to have Dr. Raktim Mukherjee with us. And we wish to say our thanks to Dr. Raktim Mukherjee that he gladly accepted our invitation and he is sparing his valuable time and expertise with all of these young learners. So sir, on behalf of Gujarat State Biotechnology Mission and N.V. Patel College of Pure and Applied Sciences, and on the behalf of all the young participants, I wish to say thank you sir for accepting our invitation, and I welcome you to take this session on animal cell culture. Over to you sir.
Thank you very much, Dr. Balani, for your wonderful introduction. And just a note of confirmation that I am audible.
Yes sir.
Okay. Now, my dear young and ignited minds of India, the budding scientists of tomorrow, myself, Dr. Raktim Mukherjee, and I am going to talk on introduction to animal cell culture. This is a very important area from research perspective, and there are huge applications of this topic. Besides that, it features in several competitive examination units such as IIT JAM, JNU Biotechnology, and of course, even at PG level for CSR UGC NET and ICMR NET. So overall, this unit is very important as far as applied life sciences is concerned. So now, since there are two issues that I discussed with Dr. Balani before I took this topic, there are students from diverse disciplines. There will be few from biotechnology, majority of them, but there will be students from other disciplines as well, mainly from botany, from zoology, from other allied areas of life sciences. So for most of you, animal cell culture, if not for biotechnology, for other students, it might be just a beginning. So I have planned to give you an overview, and only then I will get into the details. So first I will start with what is cell culture. When you talk about cell culture, it refers to the removal of cells from mammal or animal, and after you remove the cells, you have to subsequently grow the cells in favorable artificial environment. The word artificial is very important, since we are going to mimic the in vivo environment within the body of the animal into a petri plate or other allied areas. So this is what is called as cell culture. And whenever we want to remove cells, there are two ways that we can basically do. One can be by mechanical disaggregation, that is by shearing force. You just remove the cells by doing away with the extracellular matrix. Another, of course, is enzymatic disaggregation, that is, use enzyme to digest the extracellular matrix, and thereafter you can remove the cells. I know at this beginning you might not be aware of the term extracellular matrix, but of course I have those details in the succeeding part of my lecture, so I am not repeating those right now. At the end of this entire cell culture, you get what is called as an established cell line or a cell stream. Now, some of the facts about mammalian cell culture. For students who are already aware of plant cell culture, the plant cells are totipotent. You take a bird, you take a leaf, you take a meristem, a apical meristem, you take a root, you take embryo, and eventually you end up with the entire plant. This is not the case with mammalian cells, except for the blastocyst stage of embryo development, where some degree of totipotency, of course, is there. But then as mammalian cells are delicate, they are vulnerable to mechanical damage. They are present in lower growth rate, considerable yield is very less, even with lots of expertise, and you require more complex media. When I say you require more complex media, what I mean is that the animal cell culture media, the recipes are many, as well as they are complex, and for every single cell line, the media is going to be different. In addition, you require special substrates for culturing mammalian cells. Overall, to summarize this particular slide, I would add two sentences. One, technical expertise. You cannot, it's just like opening the match in a cricket. You have to open the innings from Ranji Trophy level, from your school level, university level. Only then you can open the innings like Sunil Gavaskar or Sachin Tendulkar, right? Jeffrey Boycott, some of the leading opening batsmen of all-time best in the cricket. So here also you need technical expertise. You have to learn the technique. You need to know the troubleshooting, and along with that, your wallet or your research funds should be adequate, because mammalian cell culture is definitely going to be a time as well as budget-intensive process. As I said, since you are undergraduate students, I will start with an overview and then I will go to the detail. So when I start with the overview, first I will talk about primary culture, how exactly you isolate the explant. Disaggregation, of course, I thought, that is removal of extracellular matrix. Two possible ways: mechanical disaggregation and enzymatic disaggregation. Again, during enzymatic disaggregation, you can prefer to add PDTA. You need to have different culture media depending on the cell line. You have to maintain the physiological condition, and finally, before senescence or the plateau phase is attained, you have to go for subculturing. I know at this stage it might be a bit of Greek and Latin for most of you, those who have not been acquainted to animal cell culture, but I promise in the very next slide I will make things crystal clear before going into the advanced area of my lecture. So there it is. Entire next slide I have put it in animated format. There might be slight lag in the YouTube channel that is being played, and for this reason I am deliberately pausing for some time so that entire audience can get synced with me. So here I start. First we start with live animal. When I start with live animal, I refer to this as an entire organism. So we start with organism, and then going by the cell biology textbooks, I will start scaling down. That is organism to organ system to organ. From organ I will go to tissue. From tissue I will remove the extracellular matrix, and after I remove the extracellular matrix, I will be ending up with cells. So I hope by now the YouTube viewers might have already got a taste of the slide, live animal. And this live animal, I will dissect this live animal. Okay. I'll dissect this live animal. So once I dissect this live animal, it will enable me to remove the tissue. This step is called as isolation of explant. That is, whichever tissue of my interest, I'll be removing this. Say for example, in the current slide or example, I am interested in removing the liver. So this was the liver, and I have isolated this explant from the dissected animal. So now I have the liver. You know, liver is an organ. Now what next I am doing? I am going to mince the liver tissue. I have used the analogy of a Caesar, but of course we are going to use a series of scalpels and the scrapers to mince this particular liver tissue. Once I have minced the liver tissue, now I have fragments of this liver tissue. Then I get cells inoculated in fresh culture media. But of course, this will be with debris. This will be with extracellular matrix. I just want cells. I don't want all those debris. Therefore, what I have to do, I go for cell culture. So when I go for cell culture, I digest with proteolytic enzyme. When I say proteolytic enzyme, it might be trypsin, it might be collagenase, or hyaluronidase, or any other proteolytic enzyme. I will be digesting the minced tissue with proteolytic enzyme. And this digestion of proteolytic enzyme is a very important step. What it does, it digests the extracellular matrix, and I am just left with the cells. Actually, my aim was just to get cells because the title of my talk is Animal Cell Culture. Thereafter, I will go for subculture or passaging. Once I go for subculture or passaging, I will go end up with the cell of my interest. So this is in nutshell or overview of what exactly I am going to talk during the tenure of my lecture. I start with a live animal, dissect it, remove the explant or tissue, mince the tissue, digest it with proteolytic enzyme, and thereafter I go for subculture or passaging. I end up with the desired interest, that is the cell which I am interested. This of course is also the primary culture in a nutshell. So this gives my viewers, the students, an overview of the entire process, what exactly animal cell culture is all about, or what we are going to study in detail during the succeeding tenure of our lecture. Now I am concentrating on some of the typical growth condition, and this is a very simple area, but often students fail in answering the question. And the biggest problem for this area is whenever you talk about a device or gadget, we will come to it in succeeding slide. The name of the instrument is called as CO2 incubator. Since the name of the instrument is CO2 incubator, most students uh, understand or comprehend in the mind that since the name of the instrument is CO2 incubator, it is carbon dioxide which will be required in more percentage. But actually, this is the area of concern. You remember, students, the temperature for mammalian cell culture needs to be 37 degrees centigrade. The carbon dioxide should be 5%. I will just elaborate that it should be 5% CO2 and 95% O2. But unfortunately, during entrance examination, it becomes the other way around. The name of the device is CO2 incubator. So students write 95% CO2 and 5% O2. It should be 5% CO2 and 95% O2. You have to, it's also called as humidified moist chamber. And why another name for CO2 incubator is humidified chamber? Because this instrument also maintains the relative humidity, which in case of mammalian cell culture should be 95%. So once again, I repeat for the benefit of viewers, 37 degrees centigrade temperature, 5% CO2, and relative humidity 95%. So these are the typical growth conditions for the mammalian cell culture. Now, as I said, I just zeroed in on CO2 incubator. These are some of the major equipments used in animal cell culture. You have to work on laminar air flow hood, which has a HEPA filter. And this is the area where you are doing all sorts of your culture steps such as passaging, the trypsinization, subculturing, and related steps. You have to view live cells, and so that you view live cells, you have to you cannot work with ordinary microscope. You have to use inverted microscope. The inverted microscope helps in direct visualization of cells. And if you see the diagram which I have projected, you have a light source both from the top, and there are objectives on the center. And this is the viewing area where you view live cells. So you place your Petri plate onto the stage. So this is a special kind of microscope with a light source from the top. This is called as an inverted microscope. Another gadget which you require, of course, we talked, is CO2 incubator. This device maintains carbon dioxide, which is 5%, because this gadget is connected with a carbon dioxide cylinder, and 95% O2. In addition, after the entire culture step is complete, you need to store the cell culture, whatever cells you are culturing, and for that you require cryo container. Essentially, cryo containers uses liquid nitrogen. Again, one more extra information for the benefit of viewers. We have neighboring Vadodara, within our very own Gujarat, Inox company from, not Inox movie theater, of course. Inox Vadodara is the leading person for manufacture of cryo container. Okay. Next segment of my lecture, of course, will focus on the difference between in vivo versus in vitro system. And I have placed it in an animated GIF format. On the left panel, you can see the in vitro system. In fact, whatever I have used is a live beating cardiomyocyte. And on the right panel, I have animated of in vivo, that is the rat or the mouse model. So this is the different. Next, we'll be talking about the in vivo versus in vitro system. Okay, so what's the basic difference between in vivo and an in vitro system that we are going to discuss in the next segment of our lecture? First, we will talk about the advantages of in vivo versus in vitro system. In case of in vitro system, the experiments are controllable. You can replicate the experiments over and again, and you will end up getting a good or desired result. The experiments are repeatable. There are rapid assessment, and of course, it is inexpensive. So, in vitro model is relatively inexpensive as compared to the in vivo model. Then the experiments are simple. It decreases the use of animals. This is a very important point because there are lots of ethical issues with in vivo experiments. You don't get animals that easily, and therefore it decreases the use of animal in case of in vitro experiment. You have a very good quality control. Since you can replicate the results, the quality control is very good, and this is the reason for reproducibility, or you get good results over the period of time. The range of application is vast. There are lots of application of in vitro experiments or cell culture. Of course, I have a separate slide to depict the application part of animal cell culture. I will talk in those during that duration. Whereas in case of in vivo, the advantage is it's a complete physiological response. When you talk about the complete physiological response, we have all the organs and organ systems intact, and therefore it can simulate a real body condition. It's a clinically relevant model. When we say clinically relevant model, in vivo is exactly the original model of the experimental setup. In vitro can never give the actual results of in vivo experiment. And when we work with in vivo model, we always have an infusion of immune and inflammatory response. This infusion of immune and inflammatory response is very important because when you are working with in vitro, you are just left with the cells, nothing else. You are just left with the cells, and therefore you need to include the immune and inflammatory response because in real time your cells are not alone. Your cells are continuously talking with the neighboring cells, and it is embedded within the tissue. So lots of social interaction within the cell, which is not possible in vitro condition. These were some of the advantages. Now I will move to some of the disadvantages of in vivo versus in vitro system. So what are the disadvantages of in vivo versus in vitro system? The first disadvantage of in vitro system is extrapolation of results. Whenever you talk about the extrapolation of results, in vitro system is very difficult to extrapolate to human system. Why it becomes difficult? Because there are dosimetry concerns. The areas of concerns are, one of the area is several organ system interaction is missing, and the metabolic rate of the test compound is significantly less in vitro condition as compared to in vivo. Another disadvantage is chronic effects cannot be tested. Over a long period of time, what are the chronic effects you cannot test because the blood vascular system is not present, the nervous system is not present, the excretory system is not present. Anything and everything that you are doing is just by playing with the media, and these are the disadvantages of in vitro system. What are the disadvantages of in vivo system? One disadvantage is in vivo system is expensive. It's very expensive because lots of animal, you have to feed the animal, you have to keep an animal house attendant. It is time consuming, laborious, because animal experiments are time consuming, laborious. It takes duration. Say for example, spermatogenesis, you have to maintain the animals for 45 to 60 days because that is the duration of rat spermatogenesis. You have to have ethical and regulatory issues. There are lots of regulatory issues. In fact, I will share my own experience. When I went for training in Indian Institute of Chemical Biology, Kolkata, even for training, they were not ready to give animals that easily. Lots of ethical issues are there. So looking to all these advantages and disadvantages, of course, in vitro, though with some of the disadvantages, is accepted now, at least for the initial part of the experiments as compared to in vivo. Of course, as a scientist, you need to do both in vitro as well as in vivo experiments. So to look into the disadvantage, after looking into the disadvantage and advantage of the in vivo and in vivo experiments, this is a way out. This is called as integrated discrete multiple organ co-culture. What exactly it is? This is basically a transition between an in vivo and in vitro. We just thought in vitro system can never give you the correct picture. So what best you can do is you can just replicate near in vivo system despite remaining in vitro system. This is called as integrated discrete multiple organ culture, IDMOC. And what this IDMOC has, IDMOC, you have a different plates. You can plate organ A in say hepatocyte or liver. You have another well where you can place kidney. You have third well where you are placing vascular endothelium. So what happens in our body? In our body, any response or in vivo system, any response that you study is not isolated. It is always a merging of various confounding factors. Say the blood is traveling or connecting the entire tissues, the only liquid connective tissue, the blood. You have not just one organ, you have an array of organs. You have the metabolic central command. These aspects are missing as far as the in vitro system is concerned, and therefore you have to mimic those conditions, and one of the ways integrated discrete multiple organ culture. And finally, how you are connecting all the organs or all the cells together. Here where my cursor is pointing, you have an overlay medium. This overlay medium, you are placing overlay medium which is connecting the well A, well B, and well C. So despite being in cell culture or in vitro system, you get a real life feel of in vivo culture. So this is a very popular experimental model. Okay. Now, as I said, one of the applications of cell culture is an excellent model system. Normal physiology and biochemistry of cells can be studied using cell culture. You can study the effect of various drugs and toxic compounds using animal cell culture. Some of the prominent phenomena such as mutagenesis and carcinogenesis can be studied. It can be used in drug screening and development, as well as large-scale manufacture of some of the biological compounds such as vaccines, insulin, therapeutic proteins, as well as interferons. I will add something extra to this slide, especially in case of targeted cancer therapy. You are screening, chemotherapy. You give chemotherapeutic drugs to cancer patients. Cisplatin is there, deoxyribosin is there, but most of the chemotherapeutic compounds are highly cardiotoxic. Again, there's a problem that after spending some 20,000-30,000 rupees, the chemotherapeutic compound, you find that the chemotherapy is not responding to the type of cancer. So you end up spending some 20,000-30,000 rupees, and after that, you get a negative result. Instead, when you can carry out a in vitro experiment and test whether your chemotherapeutic compound is really acting on your cancer cell, and after that, you can start your cancer chemotherapy. So this is a very important application of animal cell culture. And in Indian rupees, I will tell you the cost implication. If a chemotherapy cycle costs 30,000 rupees for a single cycle, targeted chemotherapy using in vitro system is just 5,000 rupees. So using 5,000 rupees or investing 5,000 rupees, you get to know whether your chemotherapy drug is really acting or not. So this is a diagrammatic overview of the various animal cell culture applications. You have vaccines. There is lots of funds for vaccines for COVID-19. So polio vaccine, measles vaccine, rabies vaccine, these are some of the examples. The list is exhaustive. Vaccines are a product of animal cell culture. Some of the hormones, especially human growth hormones, somatotropin, the follicle stimulating hormone, human chorionic gonadotropin. These are some of the hormones which are produced by using animal cell culture technique. Then, of course, monoclonal antibodies, anti-lipopolysaccharide, human B-cell lymphomas, antifibrin, as well as cancer immunotherapy for cancer. The checkpoint inhibitors, those monoclonal antibodies are also produced through animal cell culture. You have immunobiological regulators such as erythropoietin, often used in case of kidney or renal failure patients. Erythropoietin helps in erythropoiesis, or the buildup of your RBCs. Interleukin 2, tumor necrosis factor, and granulocyte stimulating factor. These are some of the immunobiological regulators. Some of the clotting factors are also produced from animal cell culture, such as factor 7, 8, 9, 10. Of course, some general knowledge along with my lecture. Factor 13 is also called as the Christmas factor. Interferons, interferon alpha, beta, and gamma. You might be already aware that these interferons have very important antiviral role. You have plasminogen activators such as tissue type plasminogen activators, urokinase 5, and recombinant plasminogen activators. Whenever a patient suffers from myocardial infarction, these plasminogen activators are used. Very good example which I can give to the budding undergraduate students is streptokinase. Whenever you have a myocardial infarction, you often hear that in colloquial or vernacular language, people say that doctor है तो एक 50,000 के 30,000 नो injection inject कर you. So what exactly they are doing? They are using streptokinase, and these plasminogen activators, they will dissolve the blood clot and resume the blood circulation which has been blocked in the coronary arteries. So these are some of the applications of animal cell culture. When I go further, there are certain factors that affect the behavior in complex in vivo environment. The local microenvironment, the metabolites, the local growth factors, the extracellular matrix, the architecture, the cell-cell interactions, the circulating proteins, the cytokines, the hormones. These are all the factors which affect the complex behavior in vivo environment. Now, as a sure shot reality, all these things we are not getting when you are culturing cells in the in vitro condition. So, how best to mimic this in vitro condition? Okay. How best you can mimic under in vitro condition, that is the real area of concern. And this will lead us to the further or the third segment of our lecture, that what are the methodology adapted, or how exactly you should practice animal cell culture under laboratory condition. So what we do to mimic this under in vitro condition? For this purpose, first I will take you to two important terms. One important term based on the morphology of cell is anchorage dependent. Anchorage dependent in colloquial Hindi or national language, I will say, पैर के नीचे जमीन होना, or adherent cell lines. You need a substratum or a base for adherent cell line. Likewise, in simple language, suspension culture means floating on the medium, or पैर के नीचे जमीन ना होना, or you don't have the substrate. So those are the diagrammatic areas. Now I will go to the text. Anchorage dependent is also called as adherent. Adherent is one which sticks to the surface, or it is also called as monolayer culture. So adherent culture or monolayer culture. And what happens is, the bottom of the culture vessel is covered with continuous layer of cell. You can see the bottom of the culture vessel, and this bottom of the culture vessel is covered with a continuous layer of cells. This is the monolayer culture. Some of the examples are MCF7, PC3, Of course, this examples we will again revisit when I talk about the various cell lines in the last part of my lecture. Likewise, anchorage independent पैर के नीचे जमीन ना होना, or also called as suspension culture. Suspension culture, you have majority of continuous cell line which grow as monolayers. Some of the cells which are non-adhesive, examples of leukemia, the blood cancer, some of the cells can be mechanically kept in suspension, and this is where it is. You can see, compared to the adherent cells, the cells are not attached to the substratum. This is also called as the suspension cells. So suspension cells keep floating in the medium as compared to adherent cells, which are generally suspended. Adherent cells stick to the surface, whereas the non-adherent cells or suspension cells keep floating in the media. Example is K562, whereas example of anchorage dependent is MCF7 and PC3. Now we talk about the culture surface. Most adherent cells require attachment to proliferate, and the change in charge of the surface is important. And what we do, you can just take ordinary Petri plate and coat the surface with polyl lysine. When you coat the surface with polyl lysine, you end up getting a charged surface. Or you can always add with the coating with matrix proteins. The matrix proteins can be collagen, the laminin, the gelatin, and fibronectin. So students, there is a difference between the Petri plate that you use for normal experiments and that of animal cell culture. The Petri plates which you use for animal cell culture, they are coated with matrix proteins such as collagen, laminin, gelatin, fibronectin, or most easily with polyl lysine. And these are all charged surface, and these charged surface is required for adherence of what I talked just now, the adherent cell lines, or also called as the monolayer culture, or simply called as anchorage dependent cells. So culture surface is very important. Without good culture surface, the adherent cell lines won't stick to the surface of the Petri plate. Now let's come to some of the questions. You can get ready with your chat box. One of the following is not true for in vivo system. Question was asked from JNU Biotech Entrance in 2004. There are 4 options. Maintenance is expensive. Experiments are time consuming. It decreases the use of animal. We can simulate real body condition. Something which I have already covered during the course of my lecture. So let's see. Okay, responses have started coming. I will, without any time constraint, I'll just focus on the correct answer. The correct answer is option C. One of the following is not true for in vivo system. In vivo system does not decrease the use of animal, rather it increases the use of animal. So option C is the correct answer. One of the following is true for suspension cell culture. A does not require enzymatic or mechanical dissociation. B requires periodic passaging but allows easy visual inspection under inverted microscope. Growth is limited by surface area, which may limit product yields. And option D is appropriate for most cell types, including the primary cultures. Okay. Well, people are trying. This is a very encouraging sign that students are really putting in their report. Nothing can be better for a teacher than the visual glimpse of active learners. Well, the correct answer, yes, most of you have got it correct. The correct answer is option A, that does not require enzymatic or mechanical dissociation. suspension culture does not require enzymatic or mechanical dissociation. Enzymatic or mechanical dissociation is the property of adherent monolayer or anchorage dependent cells. We continue with the lecture. Next segment is on defined media and supplements. Now, of course, I know lunchtime is far away, and but still, I would like to start this segment with this example. On top, you can see paneer cubes. On the bottom, various paneer dishes. I am not concerned about the paneer dishes. That is not the question at the moment. I just want to tell the students that this is green gravy, paneer dish with green gravy. This is a paneer dish with yellow gravy, a paneer dish with red gravy, and a paneer dish with brown gravy. Of course, you might have tasted all the four during some of the popular restaurants that you have visited. The idea that I am trying to give to the students is that you start with a particular media, but depending on which cell type you are culturing, the media needs to be varied. These are all paneer dishes, but then you have a green, yellow, red, and a brown gravy. Likewise, for every different cell line, the culture media is going to be different. This is what I am trying to put before I advance into the further course of my lecture. So in animal cell culture, for every different cell line, the media is tailored, or the media is tailored to suit the particular cell line. So I started the media formulation. The initial studies were body fluids, very primitive. The plasma, lymph, serum, tissue extracts. In simple language, the chutney was used as media formulation because they thought that as long as the in vivo system is there, the animals always are within the natural microenvironment of the body. So why not use the same natural microenvironment of the body for animal cell culture? So they use plasma, lymph, serum, and tissue extracts. Then research advanced, and they got some idea that what are the important ingredients in the media, and thereafter we got the early basal media, which had the salts, amino acids, sugars, and supplemented with vitamins and serum. More recently, we have a defined media which are cell specific, and these media are extremely complex. When I say extremely complex, I mean for every different cell line, the media is, or the recipe is different. So this is the point where I started the previous slide. Along with the recipe, you add serum. So different kinds of media along with serum. So this was the purpose behind my previous slide, that though you have a common paneer as a menu, but depending on the various menus that you are selecting from the menu card, the preparation is going to be different. Here also, depending on the cell line, the preparation is going to be different. What are the media formulations? We just thought. Now, how exactly do you go for development of media? How exactly you go for development of media? First, we go ahead with the initial media that were the simple homogenate of living tissues or tissue extracts. But then, as the subject on animal cell culture progressed, there was a need for large amount of chemical defined media. So all these primitive media or the chutneys or the homogenate did not serve the purpose. So an entire industry or the animal cell culture industry that it gave rise to, these industries provide or manufacture the media. And in some of the popular brands available in India, one popular one is High Media, of course. They manufacture lots of media. We have the Sigma, the Fluka, okay, the various international brands. Some of the common ways: Eagles basal medium, the 1955, followed by minimal essential media. And when you talk about serum, you add horse serum, the calf serum or human serum. These serums are added as supplementation. We have a separate section where we will talk about the advantages and disadvantages of serum, where we will understand in greater detail how important the serum supplementation is for animal cell culture. So the common media are minimal essential media, the Dalgupos modified Eagles media, Roswell Park Memorial Institute media 1640. These are used extensively for hematopoietic cells. Then serum is preferred, or serum-free media is preferred. Whenever you prefer serum-free media, you always have the advantage of minimizing infectious agent. Serum is a biological product, so whenever you use serum, there is always a problem of infection. So therefore, serum-free media is preferred to minimize the risk of infection. Now we will talk about the media formulations. I'll just first talk an overview. The media formulations, we have the inorganic ion, the trace elements, the amino acids, the vitamins, and the energy source. There are so many other components, but I have zeroed down the important components of media. Every media used in animal cell culture will definitely have these 5 components: the inorganic ions, such as the one which maintains the osmotic balance of cell volume, the trace elements which acts as cofactors for biochemical pathways. The very big examples are zinc and copper, which are cofactors for several metalloenzymes such as superoxide dismutase, amino acids which are used in protein synthesis, and glutamine, also used as a carbon source at high concentration. Vitamins, they act as metabolic coenzymes for cell replication. And when you talk about the energy source, you have to summarize. The media formulation involves components to maintain osmotic balance of the cell, components to maintain the biochemical pathways, to help in protein synthesis, as metabolic coenzymes for cell replication, and very important, you require an energy source. Glucose, of course, is the energy source which is helping in driving the entire cell culture to the desired limit. So these are some of the media formulations. Again, with example: the calcium chloride, sodium chloride, potassium chloride, bicarbonate, the magnesium sulfate, glucose, amino acids. This is the pictorial representation of Dalgupos modified Eagles media, and the same pictorial representation along with FBS when it is supplemented with fetal bovine serum. We generally have a medium supplemented with serum, or we have media without supplementation of serum. Mind you, students, serum is never added when you purchase the media from the company or the vendor. Serum has to be purchased separate, and that serum is later added to the cell culture media. Diagrammatic representation of the phosphate buffered saline, the media such as RPMI 1640 and Dalgupos modified Eagles media with high glucose and without glutamine. Various variants are available in market. So many variants are available in market. This is just a representative example. Now, when you talk about RPMI media, RPMI 1640, and MCQs are asked from this area. RPMI 1640 was developed by George E. Moore, Robert Gerner, and Addison Franklin in 1966. It is used for culture of normal and neoplastic leukocytes. I am not going into the detail, but I will just zero in on the key ingredients of RPMI media. RPMI media consists of glucose, pH indicated in the form of phenol reds, various inorganic salts, amino acids, and vitamins. And this is used in propagation of the neoplastic leukocytes as well as normal leukocytes. It supports the growth of neoplastic When I say neoplastic leukocytes, it is the blood cancer cells. The leukos is white and cytos is cells. So leukocytes is white blood corpuscles. So RPMI is used in culture of the neoplastic as well as normal leukocytes or white blood cells. Now, what are balanced salt solution? When you talk about the balanced salt solution, you have the what is balanced salt solution? Balanced salt solution is made to a physiological pH and isotonic salt concentration. Isotonic, the term suggests it is at par with the normal osmolarity of the organism. Some of the solutions which are most commonly used include the sodium, potassium, calcium, magnesium, and fluoride. And students, you might be aware, isotonic solution. Whenever you have visited clinic, you might have seen that in clinic, uh, sometimes along with the injection vial, another vial or a bottle is added. What that vial or bottle contains, that vial or bottle contains what is called as the sterile distilled water, or in majority of the cases, isotonic solution. That is, in case of mammal, the isotonic solution happens to be 0.9% saline. That is 0.9 gram of NaCl to every 100 gram of distilled water. So the solutions are commonly include sodium, potassium, calcium, magnesium, and fluoride. So I have just discussed what exactly is balanced salt solution. Next, I will talk some of the examples of common balanced salt solution. Some of them are Earl's balanced salt solution (EBSS), Hank's balanced salt solution (HBSS), the glucose phosphate buffer saline (DBPS), the Ringer balanced salt solution (RBSS). Ringer's salt solution is used in isolated heart experiments where you are allowing beating heart in culture media along with cardiomyocyte culture. Phosphate buffer saline (PBS) is a very common variant used in most of the cell culture. It is universal in nature compared to the remaining four. Now, what this serum provides, we just can contend that serum provides everything. Serum provides most of the things, but whatever serum provides, everything is not defined. Still, serum for animal cell culture biologists continues to be an enigma which is beyond the scope or comprehension of the scientist. It's a basic nutrient. It contains hormones and growth factors, attachment and spreading factors, the binding proteins such as albumin, fibronectin, transferrin. Serum has natural protease inhibitors. These protease inhibitors will help in antitryptic activity or help in residual tryptic activity. And serum also helps in the maintenance of pH and buffer. Some aspect of serum there is role. Now, very important module. What are the advantages and disadvantages of serum? Serum contains various growth factors and hormones, and these serum growth factors and hormones stimulates the growth of animal cells in culture. And whenever we talk about the disadvantage of serum, it lacks uniformity in the composition of serum. Every time you purchase serum, the batch number varies. There is a batch-to-batch variability. So every time you carry out experiments using serum, the batch-to-batch variation has to be nullified by continuous experiments with every batch of serum. Serum acts as a spreading factor and contains some of the growth inhibiting factors as well. It acts as a buffer agent, which helps in maintaining the pH of culture media. And one of the major disadvantages of serum is, since serum is a biological product, this serum increases the risk of contamination. So there's a huge risk of contamination. So students, what exactly happens? Generally, there is a collaborative research. In collaborative research, what happens? Two scientists from neighboring countries or from different distinct part of the world might be experimenting on a common topic. And whenever you use a serum-based media, you definitely have a problem in sending cell line from one country to another. And why this is a constraint is because whenever you are using serum, it increases the risk of contamination. So there are several legal restrictions or cargo restrictions with transport of, as such there is a restriction. Whenever you talk about biological product, there is always a restriction in transport. Moreover, if it is a serum-based culture, because of risk of contamination, that is always a major restriction for collaborative research. Serum also functions as binding agent, and the presence of serum in media might interfere with the purification and isolation of cell culture product. And this is the reason for exhaustive industrial application, we don't use serum. We generally go for serum-free media. But of course, research for serum-free media is not that developed. We still have lots to do. And serum is so natural that till today, we don't have a complete substitute to available serum. And what serum does, it minimizes the mechanical damage or damage caused by viscosity. Just like your gearbox in your vehicle, the kinetic or the two-wheeler, the serum minimizes the mechanical damage. So whenever you are using a serum-based culture, the mechanical damage to the cell can be minimized. That is the advantage. But disadvantage, as I said, major disadvantage is risk of contamination and interference of serum with some of the cell culture products. So what is complete media? Complete media has all constituents such as the glutamine, the growth supplements, the serum, growth factors and hormones, everything put together, what we say as a cocktail is essentially a complete media. So actually, when you are working in the lab, what we have to do, we have to first do literature survey. After literature survey, we purchase serum, we purchase media, and once the cells are cultured in the media, we supplement serum. Whenever we talk about serum, generally the concentration of serum is 5% to 20%. And students, please remember, one of the reasons that animal cell culture, especially mammalian cell culture, the cost is huge because of the cost of serum. The cost of serum is considerably high as compared to the cost of the media. Now this is a very important slide. I will keep it focused for considerable amount of duration. These are the media for animal cell culture. What are the constituents or components of media and what are the purpose? We begin with common salt of sodium chloride, which adjusts osmotic pressure. Inorganic salts maintain electrolyte balance. The sodium bicarbonate, which is helping in buffering capacity because the pH of blood is 7.4. Most of the mammalian cells, the normal pH is 7.4. You have glucose, 1 gram per liter, which is a source of energy or carbon source. The amino acids, which is a source of nitrogen. You have vitamins, which act as cofactors. The growth factors and hormones, which are growth stimulators. Once again, I would like to add, whenever you talk about growth factors and hormones, we always have insulin-like growth factor, which helps in growth stimulator. And sometimes, even if a patient manages diabetes with oral hypoglycemic agents, the clinician will prescribe insulin. One of the reasons why they prescribe insulin is because insulin acts as a growth stimulator. Antibiotics for preventing contamination by microorganisms. You have phenol red, which acts as a visual pH indicator, and animal serum, as I said, 1 to 20%, which enhances growth and attachment. And I just discussed the advantages and disadvantages of the serum and serum-free media. pH is very important. Just we discussed 7.4 is the optimum pH. But then, as a scientist, how do I know that my cells are cultured in right pH? And how do I know that pH of my media is changing? So what best I can do to understand this? For most cells, the pH is 7.4. And in the media, so most of the media photographs that I depicted on my PowerPoint slide, majority of them were red. One of the reasons why most of the media appeared red, because in each of these media, we have phenol red, which is used as a pH indicator. This phenol red, it turns purple at pH more than 7.8, whereas at pH less than 6.5, it turns yellow. This is the common equation governing the control of pH. H2O plus CO2 reversibly reacts to give H2CO3, which dissociates to give H plus and HCO3 minus. To compensate this, we add sodium bicarbonate at 4 millimolar concentration to continuously maintain the pH in the culture media. Because as you start growing the cells will utilize the media, and after it utilizes the media, the metabolic waste will be generated, and it will utilize the oxygen, and this metabolic waste will lead to the change in pH, and that is the reason that pH changes if you don't subculture the cells at regular interval. This is a diagrammatic overview. At pH 4 it is yellow or acidic pH. Alkaline pH it is purple. This is phenol red, and this is the degree or gradation of color change as you play with the pH, either experimentally or naturally, if you are maintaining the cells under culture. So the concept of phenol red is depicted in the slide. Phenol red turns yellow under acidic condition and purple or pink under alkaline condition. So some of the questions related, again get ready. Below pH 6.8 phenol red indicator appears. I mean, this was fairly simple, I won't give much time. This is yellow. Below pH means you are going towards the acidic side. One of the following media component adjusts osmotic pressure. Here also we talked a couple of minutes before. The answer is yes. Okay, the correct answer, many of you have got it correct. The correct answer is sodium chloride. Sodium chloride is the correct answer. One of the media component that adjusts osmotic pressure is sodium chloride, helps in maintenance of the osmotic pressure of the media. Now, why osmotic pressure of the media has to be maintained? Osmotic pressure of the media has to be maintained because if the osmotic pressure is not maintained, the cell will get shrunken. In other words, it will become like a copper, or it will get shrunken, or it will swell and it is burst. You might have already studied the RBC experiments in your 12th standard biology or even in BSc, that unless you place the RBCs in isotonic media, if you place it in hypertonic or hypotonic, either they will swell or they will swell and burst, or it will become crenated. So sodium chloride does the osmotic balance of the media. Now we go to the introduction of establishment of primary culture. Of course, in the very beginning of my lecture, I gave an overview that how exactly the primary cell culture needs to be done. And now I will talk about this in a greater depth or detail. So before we go to the primary culture, I will explain the concept of contact inhibition. You have a normal cell or mitotically active cell keeps dividing. The mitotically active cells keep dividing. Once the mitotically active cells keep dividing, it will occupy the entire area of the Petri plate, and this phenomenon of occupation of entire area of the Petri plate is called as attainment of confluence. Once normal cell attains confluence, the growth stops. This is called as contact inhibition. This is called as contact inhibition. You further have transform cells. Transform cells or cancer cells do not show this property of contact inhibition. Rather, it can slide over one another. It can slide over multiple layers. This is called as inhibition of contact inhibition. So majority of the cancer cells or transform cells do not stop their growth. They keep on piling. This is called as inhibition of contact inhibition. So these were the concepts of contact inhibition. Why it is important? Because we are going to use this in primary culture. Students, our idea is we should not allow cells to attain confluence. If you allow cells to attain confluence, it will go towards senescence. The last part of my talk, I will talk about the growth curve, that is senescence. So you don't allow confluence to attain. Before confluence is attained, you always start with subculture. So now I am going to the original protocol. This is a slide of removal of plaster of brick wall. Our cells are the bricks. Our plaster is the extracellular matrix. So our idea is just to get these bricks or to get the cells, and to get the cells we have to digest the plaster, and one of the way to digest this plaster is to digest this plaster with the help of enzymes or disaggregating enzymes. So now I am coming to my in-depth explanation. You can have a normal tissue or you can have a tumor tissue. You already have the plaster, you have to remove the plaster or you have to remove the extracellular matrix. Therefore, you have to mince the tissue. You have to suspend in enzymes such as collagenase. After you suspend in collagenase, your entire extracellular matrix is digested. After that, you filter the cells and centrifuge. You get the initiation of medium in 225 flask. These are, of course, the primary cells. In mechanical disaggregation, you mince in fragments and thereafter you can go for resuspension. In primary explants, when you are taking from the tumor tissue, again you mince in fragments, but after that the cells start to grow, and once the cells start growth, you have to go for subculture. So these are the three areas. Enzymatic digestion. Enzymatic digestion is you digest the matrix with enzyme. Mechanical disaggregation, as analogy example, you use a chini hathodi or what is called as scraper, or that is mechanically disaggregating the cells. Primary explants, you mince in fragments, and after that you grow the cells, and you grow the cells after initiation then in culture of the culture media at 37 degrees centigrade. So these were some of the overview of the protocol that we studied in as an outline in the initial part. This is a better explanation, and the figure is relatively magnified. This is journal of visual experiments that I have taken. You take an organism, say for example fish. I am interested in cell culture of pituitary gland, that is pituitary gland I am dissecting. After dissecting the pituitary gland, I am digesting the pituitary gland using enzyme, maybe collagenase or trypsin. Thereafter, I am doing mechanical dissociation of the digested tissue. Then I am centrifuging or undergoing centrifugation. And once the centrifugation step is complete, I get the cell of my interest. The cell of my interest in this case is pituitary cell from fish, and thereafter you go for plating of cells. And you keep on plating the cells till they reach confluence. As I said, you should never allow confluence to attain. Before the cells attain 100% confluence, you go for subculture. You go for subculture. Again, normal cell, that is from organ. Isolation by enzymatic mechanical action, you culture it. Confluent, as I said, confluent, entire petri plate is occupied this cell. Rather than that, you go for subculture. And once you go for subculture, you can go for series of subculture. Each time you subculture, you end up ultimately with a continuous cell division with finite cell divisions. That is, eventually the cell is going to attain senescence. Whereas when you are culturing a cancer cell or you are culturing cells from a tumor, same step of transform cells with subculture, but here you attain the cell division up to indefinite division. That is, this is called as immortal cell lines. So this is called as mortal cell line, and this is called as a immortal cell line. Immortal cell lines never undergo senescence. So I am just coming to the last or terminal part of the lecture. Some of the common enzymes are trypsin, collagenase, heparinase, hyaluronidase, pronase, displases. Whenever you use enzymatic disaggregation, you always get considerably higher number of cells as compared to mechanical disaggregation. Why mechanical disaggregation do not give you considerable high yield of cell? Because during mechanical disaggregation, the shearing force might damage so many of your cells. Trypsinization step, you get two protocols. One trypsin at 37 degrees centigrade neutralized with serum, and cold trypsin. Here it is initially at 4 degrees centigrade, and after that you maintain the temperature at 37 degrees centigrade. Since you begin trypsinization at low temperature, this protocol is called as cold trypsin. Since the protocol begins at 37 degrees centigrade, this is called as warm trypsin. So trypsin is a very important enzyme for disaggregation. Two variants of trypsinization is available. One is warm trypsin, another is cold trypsin. So trypsinization, culturing, you get formation of primary culture, and primary culture you go for secondary cell line, the tertiary cell line, and since these are finite cell line, up to certain generation it will go, 50 or 70 generation. After that, even if you subculture, it won't go. So this is a animal cell culture for normal cell. Whereas when you culture transform cell, this is a transform cell, cancer cell. In cancer cell, with the primary cell line, you go to secondary cell line, the tertiary cell line, and a series of subculture. But it never undergoes senescence. It keeps on growing, growing and growing. This is called as cell division, which continues up to infinite division. Most of your cancer cells are have the capacity for infinite cell division, or they are called as the continuous cell lines. And why they are continuous cell line? They don't undergo senescence after this 50 or 70 generations that normal cells do. Some questions based on whatever module we covered in the last 20 minutes. Trypsin is inhibited by serum that provides the divalent cations such as you have zinc and copper, calcium and magnesium, sodium and potassium, calcium and zinc. So what might be the likely answer? Okay, you can write in your chat box. Yes, answers have started. Well, the correct answer is answer B, calcium and magnesium. That is, those are the divalent cations that provide the trypsin inhibits by the serum inhibits the tryptic activity. Whenever we discuss advantages of serum, this is one of the major advantages. Even after you do so many centrifugation, small trace amount of trypsin is always remains, and this trace amount of trypsin leads to residual tryptic activity, that is digestion of the cells which are not supposed to be digested. Whenever you are using serum, this residual tryptic activity can be stopped, and therefore the calcium and magnesium present in serum, the divalent cations, they help in the inhibition of trypsin when present in serum. One of the following is not true with respect to contact inhibition. The regulatory mechanism that functions to keep cells growing, the cell-cell contact, the transformed human cell exhibit normal cellular behavior. Contact inhibited cells undergo cell cycle arrest but do not senescence, undergo senescence. The correct answer is answer C. The transformed human cells exhibit normal cellular behavior and mediate their growth and proliferation. Now, when you talk about the cell line, the primary culture is subcultured, it becomes a cell line. Subculture or passage refers to the transfer of cell from one culture vessel to another. And this we discussed earlier, that before you attain confluence, you remove some of the cells and those cells will again grow as a new culture. This way, removing certain cells and growing them over and over again is called as subculture step. Now, some important background information about continuous cell lines. You have Henrietta Lacks, cervical cancer patients from MD Cancer Research, Anderson Cancer Research Institute, and Professor George Otto Gay, who was the oncologist looking after this patient. He removed some of the cells from Henrietta Lacks and grew them in culture without informing the patient. Those days, luckily, ethical issues were not there. And to his surprise, what he found was the HeLa cell, also called named after Henrietta Lacks. So this is the immortal life of Henrietta Lacks. Henrietta Lacks died in 1930, but even today, HeLa, the first transformed cell line, we are all still using it. So it's a cervical cancer cell line. So HeLa cell is a type of immortal cell in scientific research. It's the oldest and the most commonly used human cell line, derived from Henrietta Lacks, a cancer patient who died on October 4, 1951. So this is the book review of Henrietta Lacks, Immortal Life of Henrietta Lacks. And Henrietta Lacks left, or she died or left this world, but she gifted the entire world or animal cell culture biologists with a novel cell type, that is the cell line, cervical cancer cell. What is the difference between finite and infinite cell line? Finite cell line undergo finite number of division, whereas infinite cell line, as I said, do not exhibit property of senescence. They, just like HeLa cell line is an example, they do not exhibit property of contact inhibition. And right from 1930, even today, 2020, the same HeLa cells obtained from Henrietta Lacks, we are still culturing all across the world. This suggests infinite cell lines never undergo senescence, unlike finite cell lines. Finally, the last two part of my lecture, the growth curve of animal cell. Whenever you go for animal cell, just like microbial cells, if you have studied in other subjects, initially, even the animal cells show a lag phase. This is the time when the extracellular matrix is being degraded. There is considerable lag phase where the cells are not growing. After this, there is a log phase where my cursor is pointing. This is the active cell division stage, and here the mitosis is occurring. After this, you have the green zone, the stationary phase. What is the stationary phase? The stationary phase also coincides with the contact inhibition or what is called as senescence. So generally, you don't allow senescence to take place. When you don't allow senescence to take place, what you have to do, generally just before this stationary phase, the green mark, you remove some of the cell from log phase and take it onto a separate Petri plate and start subculturing. That way, the cells won't die or you won't get senescence. So these are the phases of cell growth. Initial lag phase, cells do not divide. a log phase where the cells actively proliferate, and a exponential phase or a plateau phase which leads to senescence. Now, in your syllabus, there is a cryopreservation of cells already given. So, slight bit of cryopreservation, and after this, we'll have the closing part of this session. First of all, you passage cells. I'm just going into the steps for paucity of time. Resuspend the cells in serum containing media. Already talked about serum during the earlier course of my lecture. Then you centrifuge and aspirate the supernatant. That is, you remove the entire supernatant. You are just left with the cells. And those cells you want to cryopreserve, so that again and again we don't no need to repeat these initial steps. After this, you resuspend the cells in 10% DMSO. What is dimethyl sulfoxide? DMSO is cryoprotectant. Cryoprotectant is necessary to prevent ice crystal formation while you are freezing the cells. So you are resuspending the cells in 10% DMSO in fetal calf serum. After this, you transfer the cells to cryovial. Do not go for direct freezing at -190. Again, you will end up getting ice crystal. First, you cryopreserve at -80 degrees centigrade refrigerator, and after that, you can transfer the cells to liquid nitrogen storage. Cryopreservation is very important in your lab. It can save lots of your time. Generally, what we do before we start animal cell culture, we always cryopreserve some of the cell, because some of the experiment has got contaminated, it has gone bad. We always cryopreserve so that you can restart the experiment without waiting for cell lines to come to your lab from repository. Repositories are the place where cell lines are maintained. One such repository in India is National Center for Cell Sciences, NCCS Pune, CSR Institute, which maintains so many different cell lines, or it has a very well stocked repository. You order those cell lines, it arrives in your lab. So this is cryopreservation of cells. Now, some of the list of commonly used cell lines in culture: BHK, Syrian baby hamster kidney; CA2 is Chinese hamster ovary; HeLa, just now we talked, human cervical carcinoma; MDCK is Madden Derby canine kidney; Viro is African green monkey; and some of the continuous cell line: DU145 is prostate cancer; LMCA2 is prostate cancer again; MCF7 is breast cancer; PC3 is prostate cancer; SAUS2 is bone cancer; and CACO2 is colon cancer. These are why I have projected this slide. These are some of the experimental, whenever you do experiments, these are some of the common cell lines that most of the laboratories or scientists in India are working, those who are doing cancer research. Some of the questions, last part. One of the following is not a human prostate cancer cell line. Of course, it might be fresh in your mind. The answer is SAUS2. One of the following is a breast cancer cell line isolated in 1970 from 69-year-old women, Frances Mellon. The correct answer is MCF7, just which we projected in the previous slide. Finally, I would like to acknowledge, before ending the presentation, to Ms. Megha Dave, my student and current faculty, for helping me with the compilation of question bank. And not just a good teacher, but a very good human being and a wonderful, eloquent speaker, Dr. Amit Balani, Associate Professor from CBM University and the coordinator of the current program. He is the one who provided me this platform for delivering this talk. And of course, Dr. Anshriya Vadalkar, Joint Director, HRD GSBTM. Under aegis of GSBTM, this capacity building cell is conducting the program for our constant motivation and support. And my email ID already had projected on the first slide, bhaktimukherjee2003@gmail.com. In case of query, you can always get back to me. And as such, Amit sir might have planned something once the entire lecture session gets complete. So thank you very much. I would now hand over the proceeding to Dr. Balani.
Thank you, sir. Thanks a lot. And it is really nice. And you might have seen the text box as well.
Yes, yes, by a very interactive audience.
Yeah, so students have thoroughly enjoyed.
I give a loud round of applause, online applause, I would say, because we are dealing with high COVID times.
Yes, sir. So the most of the credit goes to this network of biotechnology capacity building cells through which now we all are connected, and we are thankful to GSBTM that particularly Dr. Subhash Soni and Dr. Anusuya Bhadalkar madam, that they have envisioned this program, and in this COVID time also they have taken utmost care that a student should not be left without any preparation. So on behalf of GSBTM sir, on behalf of NV Patel College of Pure and Applied Sciences and this NBT CBC, I wish to thank you for this entire session, and from all the participants also, it's a token of our gratitude in the form of thank you that you have spared your time, and this was a wonderful session. I have also listened to this entire program, and once again, thanks a lot, sir. Thank you for giving this particular interactive session to the students. We have also joined you in the WhatsApp group. So there might be some queries, sir, so you can solve it on WhatsApp as well.
Sure, sure. I am looking forward to the queries from students. Thank you very much, sir.
Okay, sir. Manish bhai, इनको कह दो बंद कर. Okay, sir. So within 7 to 10 minutes, we will be having another session by Dr. Dhruv Dhananjay on semiconductor and semiconductor devices. So I will tell the students that we have already sent you the link of the next lecture. So after a break of 3 to 5 minutes, please join on the another link which has been sent on the WhatsApp group, Crash Workshop 1 as well as Crash Workshop 2. Both the groups we have sent the information. So please stay tuned. So please be here.
हेलो ईश्वर भाई, अरे तू सेशन कंप्लीट થઈ ગયું હતું? मैंने आवाज़ बंद कर दी थी तब यहाँ से.
वो आना शुरू होगा.
ठीक.
वो गाना शुरू हो गया।
अच्छा उसमें यहाँ
शुरू
पे
से हुई।
मैं भी देख सकता हूँ उसमें। डॉक्टर मैं ऐसा पूछ रहा था
हाँ,
कि ये लैपटॉप किसका है दूसरा?
मेरा।
हमारा ही है ना?
हाँ,
तो मैं उनको रखूँ मेरे पास तरफ
इसलिए
एक और डेटा उसमें रखूँगा, कुछ क्वेश्चंस रखूँगा साथ में।
ऐसा है
हाँ।
क्योंकि मैं इसके
नहीं चलेगा।
फीडबैक भेजता हूँ
आई विल कीप दिस मोबाइल
फीडबैक भेजता
चलेगा,
हूँ सर इसके अंदर जो फीडबैक सेशन
डॉक्टर इट्स ओके।
आपके इसमें
नहीं कोई बात नहीं।
ना सर ना सर बुक कर दो हाँ सर हाँ हाँ
विशाल बेसो